unfold.
Welcome to the Deep Dive. For a very long time, our understanding of environmental health, it really rested on what felt like a biological guarantee. The exposure paradigm.
Right, the idea that we were separate from the pollution.
Exactly. The assumption was simple. Pollutants, whether they're heavy metals or, complex synthetic chemicals, they were external. They were outside of us.
We might breathe them in or eat them.
Sure, but our bodies, our systems, our kidneys, our liver, that have been honed over millennia, they'd process them.
They'd excrete the majority, and the core of you, your actual structure, would remain intact.
That paradigm, it gave us a real sense of control, didn't it?
It really did. It suggested that all this industrial waste was basically a filtering problem, a load our body was built to manage as long as we didn't, catastrophically overwhelm it.
The whole story was one of containment and then eventual elimination.
It goes away.
Okay, so let's unpack this with the source material we have today, because the research that's been emerging in just the last, say, 24 to 36 months.
Really, 2023, 2024, into 2025.
Right. This research demonstrates that this old paradigm isn't just challenged, it is fundamentally broken, especially when we start talking about synthetic polymers.
About plastics.
So the mission of this deep dive is to synthesize this. Just overwhelming empirical evidence showing that we are no longer merely exposed to plastic. We are structurally, intimately, and, irrevocably integrated with it.
And this is a deep dive into biological reality. The accumulation of these synthetic polymers inside human tissue, it's past a crucial inflection point.
So we're beyond the idea of contamination, like a stain you can wash out.
We are so far beyond that. We're in the realm of permanent architectural change, integration. I mean, the scientific community is now being forced to map the anatomy of a whole new biological phenotype.
One that researchers are actually calling Homo plasticus.
Plastic sapiens. It's not a metaphor anymore.
And what makes this deep dive different, I think, is that we're moving past the traditional toxicology, the focus on chemical poisoning.
Right, the chemical leaching aspect.
To really confront the physical reality of a foreign material displacing our own cells. We're going to explore the deepest sanctuaries of human anatomy, places once considered just, impregnable.
The brain, the heart, the lungs, and the reproductive system.
And the clinical descriptions we're going to share, they cross into territory that's often described as body horror.
It's an accurate term, unfortunately. We are looking at how our basic structural integrity is being compromised, how delicate cell matrices are getting scarred over, and how the physical presence of the synthetic debris, these tiny jagged shards, is mechanically sabotaging our most essential biological functions.
So we need to understand not just where the plastic is,
but what it is actively doing to our fundamental structure.
Okay, so let's start at the source of this whole conceptual collapse. The moment that boundary between the organism and its environment just vanished. For generations, every textbook drew a very clear line. The living organism on one side, the synthetic environment on the other.
Clean and simple.
But the executive summary findings in our sources, they stated unequivocally, that distinction has effectively collapsed.
It's gone.
It's no longer passive accumulation. This is an active internal colonization.
That's the perfect way to describe it. It's the difference between, say, a leaky roof where some water gets in and an active invasion of mold that has become part of the very structure of the drywall.
You can't just wipe it off.
No, it's inside. The material isn't just passing through your digestive tract. It's being bioassimilated. It's being incorporated into the cellular fabric of your body. And the very thing that makes these polymers so useful, their inertness, is what makes them such persistent and effective biological colonists.
And this bioassimilation, according to the longitudinal data we have, it seems to be accelerating exponentially.
It is, which forces us to admit that we are all, every one of us, subjects in a mass biological experiment.
An experiment where our carbon-based life is being, force hybridized?
Force hybridized with petroleum-based synthetics.
Yeah.
And you have to consider the temporal scale here.
The evolutionary mismatch.
Exactly. Human biology, through natural selection, has spent tens of millions of years fine-tuning mechanisms to interact with, to degrade, and to process organic material. Proteins, carbohydrates, even natural minerals.
Right.
Plastics, polymers like polyethylene, PVC, polypropylene. They've only been widely available since the 1950s.
So 70 years versus millions.
It's not a fair fight. Our body has no evolved toolkit, No chemical pathway, no specific enzyme to effectively deal with this high-density, high molecular weight foreign material.
In the scale of that mismatch, it explains this concept from the sources about the fibrosis we're seeing across multiple organs.
Yeah.
That it's not just random cellular breakdown.
No, not at all. It is the clinical manifestation of a desperate, failed immunological war against an indestructible invader.
A failed war.
Think about it. When your body encounters a threat, say a virus or a bacterium, the immune system launches a response. A macrophage tries to engulf it, destroy it, report back.
But when it encounters a shard of plastic,
It recognizes it as non-self. It knows it has to be eliminated.
right.
But chemically, it can't be killed. It's metabolically inert. It's designed to last forever.
So the immune system is just locked in this perpetual battle. A battle with a particle that is designed for environmental permanence.
Exactly. And the result of that perpetual, futile warfare is chronic inflammation. This leads to the body laying down massive amounts of collagen,
Scar tissue.
fibrosis. It's the body trying to wall off the invader to create a kind of biological sarcophagus for the plastic.
But in doing so, it's replacing functional tissue.
It's replacing the good stuff, the elastic, functional tissue like the filtering membranes in your liver or the gas exchange units in your lungs, with rigid, non-functional scar tissue. It's a mechanism of self-destruction, and it's driven by an external, inert threat.
The consequences of this failed war are systemic.
Yeah.
And that brings us to the first major anatomical sanctuary we need to discuss, which is, well, it's arguably the most sensitive and alarming of all.
The human brain.
Historically, in both toxicology and neuroscience, the blood-brain barrier, the BBB, has been described as the body's ultimate defense.
The impregnable fortress.
The impregnable fortress. It ensures that the brain, the actual seat of your consciousness and your identity, is ruthlessly protected from toxins and pollutants that are circulating in your blood.
And that assumption has been the bedrock for neurological safety standards for decades.
But our sources confirm that assumption has been thoroughly shattered.
The BBB is just. It's a masterpiece of evolution. It's formed by these specialized endothelial cells that line the capillaries of your central nervous system, and they're held together by these incredibly tight junctions that are almost impossible to pass.
It's a gatekeeper.
An active gatekeeper. It rigorously controls what gets through. It allows oxygen and glucose in through specific transporters while just excluding large or unwanted foreign materials. So the discovery of nanoplastics, these MMPs, penetrating this fortress, it's a complete game changer for neuroscience and for public health.
So if that barrier is designed to be so incredibly tight, how are these particles, which are essentially solid, non-biological shards, how are they making it across?
Right, it's clearly not brute force.
No, it's something more sophisticated, almost insidious, kind of biological hijacking, what's being called the Trojan Horse mechanism.
And this is where the physics and the biology of the nanoscale intersect just perfectly. We're talking about particles that are typically measuring 1 to 200 nanometers.
Incredibly small.
And crucially, they are highly hydrophobic. They repel water. So when they enter your bloodstream, which is mostly water plasma is an aqueous environment, they're thermodynamically unstable.
So they need to stabilize themselves.
Instantly. And to do that, to minimize their surface energy, they immediately start to aggregate circulating organic molecules onto their surface.
So they're cloaking themselves. In what kind of material?
Specifically, they bind to circulating proteins and lipids, most notably apolipoproteins and cholesterol. And this molecular skin that forms around them is called the biomolecular corona.
So now, when this newly coated particle reaches the BBB, it doesn't look like a foreign inorganic shard of petroleum byproduct anymore.
Exactly. It effectively mimics an essential nutrient.
It's in disguise.
It is. To the specialized receptors that sit on the BBB's endothelial cells, that plastic particle, wrapped in its apolipoprotein and cholesterol coat, looks remarkably like an essential lipid droplet.
Something the brain needs.
Something the brain desperately needs. The brain is the most lipid-rich organ in the body. It demands a constant supply of cholesterol for neuronal maintenance, for synapse function.
So the defense system, rather than fighting the particle, it's tricked into actively welcoming it.
Yeah.
Actively transporting it.
Yes. Through a process called receptor-mediated transcytosis. The barrier literally opens the door, binds the Trojan Horse particle to its surface receptors, and then actively shuttles it across the endothelial cells and into the brain parenchyma.
And the sources emphasize this isn't an accident. Certain plastics are better at this than others.
Oh, absolutely. The source material highlights that polyethylene, or PE, and polypropylene, PP, the two most common environmental plastics, show a remarkable preference for this pathway. It's because of their physical structure and their high hydrophobicity. It makes them exceptionally good at forming that crucial lipid corona.
So once the plastic is across the barrier, we then run into the second horrifying realization. The brain isn't equipped to deal with it.
It's a transition from breach to permanent trap.
Why is clearance so limited? Why can't the brain get it out?
The brain is a privileged organ in many ways, and one of them is its unique waste disposal system. We have what's called the glymphatic system. It's a network that flushes waste products out during sleep, pumping cerebrospinal fluid through the brain tissue.
Okay, so it has a cleaning system.
It does. But, and this is the critical point, this system is primarily evolved to clear soluble proteins and fluid-based waste. Things like the misfolded amyloid beta proteins that characterize Alzheimer's.
I see. It's not designed for solids.
It is fundamentally incapable of clearing solid, insoluble, particulate matter, especially not jagged, high-density polymer shards.
So if it gets in, it's not coming out. The brain just becomes an indefinite sink.
It acts as a bioaccumulator, a physical sediment trap for the debris of the technosphere. Unlike, say, the liver, which can push certain insoluble toxins into the bile duct for excretion, the brain has no effective mechanism to expel solid matter. The particle is trapped indefinitely, and it subjects the surrounding neural tissue to chronic mechanical and chemical irritation for the rest of your life.
This conceptual understanding, the Trojan horse, and then the indefinite sink, is made truly terrifying by the quantitative data. The numbers that define what some researchers are now calling the plastic cortex.
And this comes largely from a landmark 2024 study led by Dr. Matthew 𝐏𝐿𝐾𝐿.
What did the study find?
The 𝐏𝐿𝐾𝐿 study gave us the first hard quantitative data on the plastic burden in human neurological tissue. They analyzed autopsied samples, and the findings confirmed two things immediately. First, the rate of accumulation is rapid. Frighteningly rapid.
Foul brap.
Comparing the 2024 samples to those from just 2016 showed that the concentration of these micro and nanoplastics, the MNPs, in human brain tissue had increased by a staggering 50%.
50% in just 8 years.
In 8 years. And that rate perfectly mirrors the exponential increase we've seen in global plastic production.
And the second finding, it really upends the traditional toxicological wisdom, which always assumed the liver and the kidneys would bear the highest burden.
It does completely. The data showed the brain harbors significantly higher concentrations than those primary filtering organs. I mean, in some samples, the brain contained anywhere from 7 to 30 times the concentration of plastic found in the liver or the kidneys.
7 to 30 times more.
Yeah. This empirically validates that one-way valve theory. The BBB is highly effective at keeping plastics in once they've gained entry.
And when we talk about accumulation, we are not talking about trace amounts, parts per billion anymore. We're talking about actual physical displacement of neural tissue.
We have to grasp the scale of this. The sources reveal that in the most contaminated of the 2024 samples, plastic constituted approximately 0.5% of the total brain weight.
0.5%.
Now, if you consider an average brain weight of about 1,400 grams, that 0.5% translates to 7 grams of synthetic polymer, physically embedded within the organ.
7 grams. Just to contextualize that for everyone listening, that is the physical mass of a standard credit card or a plastic spoon, now incorporated into the very seat of your memory, your personality, your intelligence.
That one statistic forces you to fundamentally redefine what it means to be physically human.
And the physical form of these invaders is crucial, isn't it? The imaging shows they're not smooth little beads.
Not at all. Electron microscopy confirms these are nanoscale shard-like fragments. They're jagged. They're weathered debris, which reflects their long journey through the environment and then through the bloodstream.
So if you have these jagged shards physically present in the delicate lipid environment of a neuron, maybe embedded in the fatty myelin sheath or near a synapse, what is the implied mechanism of damage?
Well, the damage moves beyond simple chemical toxicity into outright mechanical sabotage. These shards are physically sharp. They're hypothesized to mechanically sever or disrupt axons, which are the long fibers responsible for transmitting your electrical signals.
So they could be cutting the wires.
Or they could be interrupting the integrity of the myelin sheath. That's the fatty insulation that's essential for rapid, efficient thought transmission. This physical abrasion causes inflammation and literally impedes the neurological signaling pathways.
Which brings us directly to the profound and disturbing correlation between the cerebral plasticization and neurodegenerative disease. The existential dread really sets in when we look at the link to Alzheimer's.
The data is stark. Individuals who died with a documented diagnosis of dementia, including Alzheimer's, had up to 10 times the amount of plastic found in age-matched control subjects who did not have dementia.
10 times.
So the question, of course, becomes cause and effect. Is it just that the BBB breaks down in late-stage dementia, letting more plastic flood in? That's the atrophy hypothesis.
But the other idea, the nucleation hypothesis, is far more concerning.
It is, because it suggests the plastic is the initiator of the disease process.
How would that work?
This hypothesis links the physical presence of the plastic directly to the established pathology of protein misfolding. We know Alzheimer's is driven by the pathological aggregation of proteins into amyloid beta plaques and tau tangles.
Right.
So think of it in terms of material science. If you introduce an insoluble particle, a seed crystal, into a solution that is already supersaturated with protein, that particle provides a perfect high surface area template to trigger rapid aggregation.
So the nanoplastic shard with its organic protein corona acts as the ideal nucleation site, a little scaffold that triggers the pathological misfolding and clumping of the amyloid and tau proteins around it.
The plastic could be the spark that accelerates the entire disease pathology.
And this isn't the only mechanism. There's also a link to vascular dementia.
That's right. The plastic particles aren't just depositing in the neural tissue, but in the walls of the cerebrovascular system, in the blood vessels of the brain. This can lead to mechanical blockages of microcapillaries, causing microinfarcts,
Tiny localized strokes.
tiny strokes, and chronic hypoperfusion, which means the brain is being starved of the blood supply it needs for cognitive function. So you have a brain that is simultaneously being mechanically scarred, chemically inflamed, and physically choked off by plastic.
And the finding that the highest accumulation is often localized in the frontal cortex, the region for executive function, planning, personality, decision-making, that suggests this contamination strikes at the very essence of human identity. We're not just losing function, we are replacing the biological substrate of our consciousness with synthetic debris.
The neurological invasion is unique in its horror, but the body's systemic response to plastic is universal. It's chronic, and it manifests as this pervasive internal scarring that's now defined by the clinical concept of plasticosis.
Right, and as you mentioned earlier, this term didn't originate in human medicine?
No, it was first coined after observing just extreme pathology in non-human species, specifically in the flesh-footed shearwaters on Lord Howe Island.
What was happening to them?
Chronic ingestion of sharp plastic was causing their digestive systems to lay down excessive scar tissue fibrosis, which was leading inevitably to organ failure. The body was trying to heal the constant wounds caused by these sharp, indestructible plastic shards.
But the healing process itself, the scarring, was the mechanism of death.
Exactly. That is the prototype we are now witnessing in human organs, a self-inflicted wound.
And then this brings us back to the immune system's futile struggle, what researchers are calling frustrated phagocytosis.
That phrase it perfectly captures the cellular crisis. Macrophages are our patrol cells. They're designed to engulf or phagocytose and then degrade foreign pathogens.
But these synthetic microplastic shards, they represent a complete evolutionary mismatch.
They do. They're recognized as foreign, but they're chemically non-compliant. They cannot be dissolved by human lysosomal enzymes. They can't be metabolized.
So let's detail that frustration. What's happening at the microscopic level when a macrophage tries to engage one of these fragments?
Well, if the particle is small enough to be engulfed, the macrophage will internalize it, but then it can't digest it. The particle just stays there, intact, acting as a perpetual irritant inside the cell.
That can't be good for the cell.
It often leads to the macrophage's death, a process called pyroptosis, where the cell literally ruptures, releasing the undigested plastic particle and a flood of inflammatory contents, cytokines, and enzymes back into the surrounding tissue.
Which just attracts an even more aggressive wave of immune response.
It's a cycle of cellular sacrifice for absolutely no gain.
And if the particle is too large to be fully swallowed, like some of the fibers we inhale?
The mechanism shifts. The macrophage can't engulf it, so it latches onto the surface and attempts external digestion. It secretes a concentrated bath of destructive chemicals, acid, and highly reactive enzymes in this desperate, localized attempt to dissolve the indestructible polymer.
But that acid bath doesn't just hit the plastic.
It inevitably misses. It damages the surrounding healthy, carbon-based host tissue, and that just triggers the fibrosis cascade even more.
We're seeing the physical evidence of this cellular civil war across multiple tissues, often marked by the presence of this this foreign body giant cell reaction?
The FBGC. It's the microscopic signature of unresolved chronic inflammation. When individual macrophages realize they are physically incapable of destroying or moving the invader alone, they employ this kind of emergency cellular mechanism.
What do they do?
They fuse together, sometimes dozens of them, with all their nuclei, forming a single, massive, multinucleated cell, a giant cell, designed to physically encase and quarantine the foreign material.
So finding these massive fused cells in human lung or liver or arterial tissue, that's definitive proof that the body is trying to wall off but failing to clear this synthetic debris.
It is the highest level of biological mobilization against an intractable foe. And these FBGCs, they're not passive. They are constantly churning out a powerful cocktail of pro-inflammatory cytokines, specifically interleukin-1 beta, IL-6, and tumor necrosis factor alpha.
And other damaging chemicals.
Oh yeah, and reactive oxygen species, ROS. This chemical environment is what drives the chronic disease process. It leads to the relentless accumulation of scar tissue and potentially promotes genetic damage in the neighboring cells.
And perhaps the most dire consequence of this reaction is being seen in the lungs, leading to what's been termed the concrete lung.
This is directly related to a diagnosis called idiopathic pulmonary fibrosis, or IPF. Historically, that diagnosis meant the progressive scarring and stiffening of the lungs without a known cause. Idiopathic means we don't know why.
But a key 2025 study suggests that for a significant number of these patients, the idiopathic cause is the persistent inhalation of the technosphere.
Specifically, it's linking inhaled microplastics, particularly fibers from synthetic textiles like polyethylene terephthalate PT, to the disease.
Peat fibers.
Yeah.
So from clothing, carpets.
Exactly. When these tiny 1 to 10 micron fibers lodge deep within the pulmonary interstitium, that thin space between your air sacs and your capillaries, they activate specific intracellular signaling pathways. The study highlighted the activation of AKT1 and PIM1 pathways.
Those names are very clinical. What do those pathways normally do, and how does plastic hijack them?
Normally, AKT1 and PIM1 are involved in regulating cell growth, survival, differentiation, basic wound healing and maintenance. But when they are chronically and aberrantly stimulated by the presence of a peat fiber, they trigger a cascade that leads to the excessive proliferation of fibroblasts.
And fibroblasts are the cells that produce collagen.
The scar tissue. So instead of a normal healing repair, the tissue begins rapidly laying down dense, inelastic scar tissue. The lung loses its spongy, elastic quality. It becomes stiff, inflexible, fibrotic. It's a structural change that rapidly reduces your ability to transfer oxygen,
earning it the grim moniker of the concrete lung.
We are witnessing the internal structural consequences of our clothing choices and our indoor air quality. And that chronic destructive inflammation, it now leads us directly to the cardiovascular system, where this inflammation is proving to be directly lethal.
The connection between the plastic we inhale or ingest and cardiovascular death is one of the most clinically robust and, frankly, chilling findings of the last few years.
It is. And this evidence comes from a landmark 2024 study published in the New England Journal of Medicine, a very prestigious source.
This study, Microplastics and Nanoplastics in Atheromas and Cardiovascular Events, it's monumental because it moved the whole conversation from theoretical toxicity to confirmed mortality prediction.
Right. They analyzed arterial plaque, or atheromas, specifically from the carotid arteries of patients who were having them surgically removed.
So they were looking at the literal disease structures that are responsible for major strokes and heart attacks.
And the contamination they found was nearly universal. They found detectable polyethylene, PE, in 58.4% of the patient's plaques, and polyvinyl chloride, PVC, in 12.1%.
So the plastic is no longer just a contaminant floating in the blood.
No, it is physically integrated into the architecture of the blockage that threatens your life.
And the presence of that plastic was a stunningly powerful predictor of catastrophic outcomes.
Chilling is the right word. Patients whose plaques contained detectable microplastics had an astounding 4.53 times higher risk of experiencing a major cardiovascular event.
Meaning heart attack, stroke,
or death from any cause within the next 34 months compared to patients who had plastic-free plaques.
4.5 times higher risk. How does that compare against other known severe risk factors, just so we can appreciate the magnitude?
It rivals, or in some groups, even exceeds the predictive power of severe, uncontrolled, traditional risk factors. I mean, imagine a patient who's lived with untreated chronic high blood pressure for a decade.
Or someone with severely uncontrolled type 2 diabetes.
Exactly. Detecting microplastics in their plaque gives you a similar or often greater prognostic warning of an immediate severe catastrophe. It tells us that synthetic polymers must now be counted among the most significant risk factors for cardiovascular disease, period.
So the key question becomes why? Why does the presence of these shards so dramatically increase the risk of the plaque rupturing?
It points to a two-pronged mechanism of destabilization, which creates what clinicians call hot or vulnerable plaques.
Okay, what's the first prong?
The first is purely structural. An arterial plaque only becomes lethal when its fibrous cap. That's a layer of tough connective tissue covering the fatty core when that cap ruptures.
Because when the cap breaks, the contents spill into the bloodstream.
And trigger an acute thrombus, a clot. That's the immediate cause of the heart attack or stroke.
And the plastic acts as a mechanical saboteur.
Precisely. Electron microscopy confirmed the presence of visible, jagged-edged foreign particles embedded throughout the fibrous cap material. These are literal physical defects. They act like stress risers or tiny fractures in the structure.
So the cap is just more likely to tear.
Significantly more likely to tear and rupture under the constant mechanical stress of your pulsating blood flow.
And the second prong is that inflammatory response we were just talking about.
Yes, the localized chronic inflammation is turbocharged within the plaque environment. Plaques containing plastic showed significantly higher levels of inflammatory markers, IL-6, TNF-alpha. The immune cells are focused on fighting the plastic particles embedded in the plaque wall.
And in their futile attempt to degrade the plastic.
They release enzymes, matrix metalloproteinases that don't touch the polymer, but they do actively digest and dissolve the collagen and elastin that make up the protective fibrous cap. This whole process just makes the cap thin, weak, and highly vulnerable.
So the plastic shard both physically compromises the structure and chemically incites the immune system to dissolve the structure.
It's a perfect fatal synergy.
So we have traced the final lethal journey of a synthetic fragment. It starts in the environment, it traverses the gut, enters the bloodstream, lodges itself in an artery wall, and then acts as both a mechanical defect and an inflammatory accelerant, leading directly to death.
And if that cardiovascular integration represents a threat to the individual's longevity, the findings within the reproductive system, they highlight a pervasive threat to the continuation of the species itself.
The infiltration into reproductive tissues is proving to be absolute and ubiquitous.
It is.
Let's start with the male reproductive system. The sources detail a 2024 study that analyzed human tests. What was the prevalence of contamination there?
It was 100%. Microplastics were found in every single one of the 23 human test samples that they tested.
Every single one.
And beyond that, the sheer concentration was astonishing. 330 micrograms of plastic per gram of tissue.
Now, to put that figure in perspective, this study also analyzed dog samples at the same time, didn't it?
It did. And the concentration in the human tests was nearly 3 times higher than what they found in the dogs.
3 times higher. Why?
It suggests that while the general environmental exposure is universal, the exposure vectors that are unique to our human industrial lifestyles, high consumption of processed and packaged foods, inhalation of indoor air saturated with textile fibers, are accelerating this biological integration specifically in humans.
And the implication here is direct. This saturation correlates chillingly well with the well-documented massive global decline in male fertility.
It is sperm counts.
The decline over the past 50 years.
Exactly. The study established a direct link. Higher concentrations of polyvinyl chloride, PVC, a common rigid plastic that often contains high levels of chemical additive, in the testes tissue were significantly correlated with both lower total sperm count and reduced sperm motility. This is a direct, measurable link between the accumulation of synthetic debris and the failure of basic reproductive function.
And the disruption mechanism is described as a pincer attack on fertility.
Right, which suggests that both physical and chemical toxicity are at play at the same time.
Let's break that down. What's the physical disruption?
The physical disruption is caused by nanoplastics lodging themselves within the delicate, highly organized structures of the seminiferous tubules and the Sertoli cells. These structures are the essential factories for spermatogenesis, the creation and maturation of sperm.
So the debris is gumming up the works.
It is physically displacing the necessary cellular machinery. It's mechanically impeding the precise choreography required for healthy sperm production.
And the chemical disruption. That relates to PVC acting as a carrier for its notorious plasticizers.
Yes, PVC requires large amounts of phthalates to give it flexibility. And phthalates are classic endocrine-disrupting chemicals, EDCs. They are potent anti-androgens.
So when the PVC particle is lodged in the testes,
it acts as a persistent local source, leaching phthalates directly into the surrounding tissue. These chemicals inhibit the production of testosterone by the Leydig cells, which are the primary hormonal drivers of sperm creation. So the plastic particle is a Trojan horse carrying an anti-androgen chemical bomb, disrupting hormonal signaling and physically impeding sperm creation simultaneously.
if the male reproductive system is saturated, what does this mean for the next generation? For the maternal-fetal axis, the placenta was long seen as the final, absolute barrier protecting the fetus.
That barrier has been completely overwhelmed. A 2024 study that examined 62 human placenta samples, the very organ responsible for nutrient transfer and fetal protection, found microplastics in every single sample.
Again, 100% prevalence.
100%. And again, polyethylene, the most widely produced polymer, was the most common contaminant found.
And this contamination isn't just confined to the placental tissue itself, is it?
No. The presence of plastics has been definitively confirmed in umbilical cord blood, in amniotic fluid, and even in meconium, which is the infant's first stool. This confirms that plastics are not being stopped at the barrier, they are traversing it and entering the fetal circulation. The next generation is literally developing in what you can only describe as a plastic soup.
And similar to the heart findings, there's a suggested causal link to adverse outcomes for the child.
Yes, the sources reveal a really disturbing finding regarding preterm birth. Significantly higher concentrations of microplastics were found in the placentas of infants delivered prematurely compared to those born full-term.
Now that's critical, because if plastic accumulation were just a function of time, you would expect the full-term placentas to have higher loads.
Exactly.
But the fact that the shorter-term placentas have higher loads, it suggests the accumulation is the cause of the complication, not just a consequence of the duration of the pregnancy.
Precisely. The hyperaccumulation of plastic in the placenta may be driving chronic localized inflammation and dysfunction, a form of localized plasticosis that ultimately triggers a mechanism of placental failure or inflammatory signaling that pushes the body into early labor.
The implication is just profound. We're not merely inheriting a polluted world.
No. We are constructing the next generation using cellular and endocrine systems that are already hybridized with synthetic waste. We are born pre-polluted.
Okay, so we've mapped the anatomy of Homo plasticus. Now we need to synthesize two final concepts. First, how the environment successfully delivers this immense load, and second, why the body cannot simply dispose of it.
For a long time, the public focus was so heavily on ingestion, eating plastic through contaminated seafood or packaging. And while ingestion is significant, new data suggests we have to shift our primary concern to the air we breathe.
As a potentially more insidious vector for systemic integration.
Yes.
Why is inhalation now considered the dominant, or at least the most dangerous, vector?
Two reasons. The dose is staggering, and the particle size is critical. A 2025 study used highly sensitive Raman spectroscopy to analyze indoor air, and it found that adults inhale approximately 68,000 microplastic particles per day.
68,000. That figure is 100 times higher than previous estimates.
Which just demonstrates how poor our previous detection methods were.
And the reason for that massive increase in the particle count.
Older technologies could only reliably detect particles that were larger than 10 or 20 microns. This new research can detect particles down to 1 micron, and it found that 94% of airborne plastic falls within the highly dangerous 1 to 10 micron respirable range.
And respirable means they're small enough to bypass the body's upper respiratory defenses.
Completely. They get past the nasal hairs, the mucociliary escalator, and they lodge deep in the alveolar region of the lung where gas exchange occurs. And once they're there, they are virtually impossible to cough or clear out.
And the source of this inhaled dose,
yeah,
it's not the factory chimney down the road.
No. The primary vector is the indoor environment, where most modern humans spend 90% of their time. The study found indoor concentrations of microplastics were nearly 5 times higher than outdoor concentrations.
We're surrounded by it.
We are surrounded by synthetic textiles, polyester clothing, nylon carpets, microfleece upholstery, all constantly shedding fibers into the air we just recirculate and breathe. Our convenience is polluting us from the inside out.
Okay, let's circle back for a moment to ingestion, to that common statistic we've all heard, that we eat a credit card's worth of plastic a week. The new reality of nanoplastics suggests that the mass of the plastic matters far less than the count of the particles.
That is the essential insight. 5 grams of bulk plastic, the mass of a credit card, is largely too big to cross the gut barrier. It gets safely excreted. But if you take that same 5 grams and you grind it into billions of nanoplastic particles, you have created a biological catastrophe. A study on bottled water confirmed that a single liter can contain up to 240,000 plastic particles, and 90% of those are nanoplastics.
And those nanoplastics present a dual risk.
They do. First, they dramatically increase the total surface area available for chemical leaching. So even a small mass of plastic can dump a massive dose of phthalates or bisphenols or other chemical additives into your body.
And second.
And crucially, they are small enough to pass right through the intestinal wall. They might be actively transported across via specialized microfold cells, the M cells that monitor your gut contents, or they might just slip through the tiny gaps between the intestinal cells. Either way, they gain immediate systemic entry to your bloodstream for distribution to your heart, your brain, and your testes.
This integration is so systemic, so complete, that the geological record itself will bear witness to this change. I mean, geologists already use the term technofossils to describe the permanent synthetic remnants that define our current geological age, the Plasticine.
And the ultimate technofossil is now the human skeleton. Sources confirm that plastic has been found deep within human bone marrow and within calcified bone tissue itself. The skeleton, the very structure we rely on for identity and support, is now hybridized.
How does plastic even integrate into bone structure, which is constantly undergoing turnover?
Well, bone tissue is metabolically dynamic. It's a balance between creation and destruction. You have osteoblasts, the cells that build new bone, and osteoclasts, the cells that dissolve old bone.
Right.
Nanoplastics, transported by the blood, accumulate in the bone marrow. And there, they're thought to disrupt this delicate metabolic balance. The chronic inflammatory signals, those cytokines like IL-6 and TNF-alpha released by immune cells fighting the plastics, they inhibit the activity of the osteoblasts. They slow down bone formation, and they might even increase the activity of the osteoclasts.
Which suggests that this plastic integration could contribute to diseases like osteoporosis, generalized skeletal frailty.
Future paleontologists won't just find carbon-based remains. They will find skeletal structures intrinsically marked by the chemical signature of polyethylene.
Which brings us to the final terrifying synthesis, the absolute impossibility of clearance.
Every single finding we've discussed points to one single overriding conclusion. The rate of our intake through air, water, and food far, far exceeds the body's rate of elimination.
The body has no evolutionary mechanism to break these materials down. We're dealing with biological permanence.
Exactly. We have to abandon the comforting idea that we can somehow detox this material. Chemically inert polymers like PE and PVC are resistant to human enzymes. And as a result, unlike natural toxins, which might have half-lives of hours or days, plastic particles have half-lives of decades inside human tissues.
Especially in places like the brain and the liver.
Yes. The plastic cortex is not a momentary measurement of your current exposure. It is a cumulative physiological record of a lifetime of intake. The synthetic debris you inhaled a decade ago is still lodged in your alveoli, it is still sitting in your arterial plaque, and it is still trapped in your neural tissue.
This deep dive has mapped a forced, unguided evolutionary step. Homo plasticus is not a rhetorical device anymore. It is a taxonomic descriptor for a species whose fundamental chemical composition has been irreversibly altered.
We have crossed a boundary. We are no longer purely carbon, calcium, and water. We are carbon, calcium, water, and polyethylene. This is the new definition of our biological reality.
And the ultimate horror lies in the intimacy of this integration. The plastic isn't an external threat to us. It is now structurally of us.
It is the defect in the fibrous cap that stops the heart.
It's the chemical bomb inhibiting the production of life in the reproductive system.
It's the nanoscale shard acting as a nucleation site for the proteins that steal our memories in the brain.
We created a synthetic world, and that world has successfully remodeled our internal architecture. We are the first generation of plastic sapiens.
And the physical presence of these synthetic polymers is driving chronic inflammation and mechanical failure across all our major organ systems. We designed this age of convenience using the unparalleled intelligence housed within the human brain. But now that the physical substrate of that brain, the neurons, the white matter, the lipid sheaths, is saturated with the products of that age, this ongoing bioassimilation, it just raises profound questions about the future.
If the very foundation of our intellect and our executive function is being steadily and permanently replaced by inert, jagged synthetic debris, what does this accumulation imply for the future trajectory of human cognition, decision-making, and consciousness itself?
It forces us to ask, if the very substance of our thought is plasticized, can we still think our way out of the Plasticine? Something truly worth mulling over.
Thank you for joining us for the deep dive. We'll see you next time.